Hello, everyone.
Hello.
And this is Qiming. I'm the International Marketing Director here at Cyagen. And happen to be here in town in Boston. And it's such a lovely weather.
It is, indeed.
Yeah. So alongside with me is our Senior Scientific Manager, Dr. Mark Lötzerich. Mark, you want to introduce yourself to the audience a little bit?
Sure. Hi, my name is Mark Lötzerich, like Qiming already mentioned. I'm with Cyagen for like more than 2 years now as a Senior Scientific Manager. I have a background in virology and immunology and worked in different places all over the world on innate immune sensing, viral invasion, and also CRO work on COVID and influenza in the past. So on all kinds of animal models that have been involved up to non-human primates before joining Cyagen.
Great. Really a pleasure to have you here. And here we're in Boston, and today we're going to talk about GLP-1.
Wonderful.
So, Boston has been the hub for a lot of scientific innovations, breakthroughs, and GLP-1 is just among one of them. The discovery of GLP-1 can be dated back to decades ago from Dr. Joe Haberner's group at MGH at that time. He molecularly cloned the GLP-1 gene, and you can see nowadays where we were this morning at Eli Lilly, you see a lot of multinational companies bring their innovative pipeline and as well as a strategic expansion here to Boston. So before we delve into the bolts and nuts of GLP-1, can you tell us some basic biology about this molecule?
Sure, like the GLP-1 pathway is really interesting, and you already mentioned that it's relevant for type 2 diabetes, and that's why lots of the companies targeting this pathway. So GLP-1 is a glucagon-like peptide 1. It's like this is the name of that, and it's a hormone that is usually secreted in the intestine upon food uptake, right? So, and then it has multiple effects on the glucose homeostasis in the body. On the one hand, the induction, or in this case, of secretion of insulin from pancreatic beta cells. That's really important. So it has a positive effect that is glucose driven. And then at the same time, you have suppression of glucagon release or secretion in the alpha cells. So that there are multiple effects. Then you have, of course, a stomach retention of the food that is also there. And on the central nervous system, it has an effect on the appetite that is reduced. At the same time, you have a satiety feeling, so there's less food consumption, and that has a positive effect on weight, body weight. So you probably see that from all the different new drugs, like the injectables that are established from Novo Nordisk, like semaglutide, for example, Ozempic, or from Eli Lilly. That's also another injectable that is really promising, but there's a huge pipeline of oral available small compounds that are now under testing and show some really promising results on this particular pathway. Yes.
Yeah. So as you mentioned, the GLP-1 and GLP-1 receptor axis has a pleiotropic effect on the entire body. I think originally semaglutide and tirzepatide was designed in a way to maintain blood glucose. But in the clinics, people are starting to see some of the other benefits. One thing you mentioned is the body weight. But besides that, people are also seeing multiple other things. For example, lowering cardiovascular disease risks, as well as, say, even in Alzheimer's.
It's a brain positive effect for brain health.
Right. So right now, I think the clinicians are sort of like going from the bed to the bench side and trying to understand the biology of it, because there still seems to be like some gap between the clinical benefits and the biology or the basic science. So how do you think the mouse models can help to sort of like bridge the gap here?
Yeah, there are different options. Like for for example, now for us or for this particular interaction, the GLP-1 and GLP-1 receptor, in this case, the pathway, it is really relevant that we can use the mice in different ways. So we have knockout models or CKO models to study the basic function of this receptor. So in absence of this receptor, there should be an impaired signaling. And you can measure, in this case, glucose levels, insulin levels, and also the onset of diabetes, type 2 diabetes, also the body weight in absence of this receptor. On top of that, of course, you can, all your small compounds, your drugs, you can test that on such KO models or CKO models, unusable models, to test side effects of the system, of the drug, right? In absence of the receptor where it should bind, because the drugs are, in this case, agonists, receptor agonists that induce signaling in absence of the GLP-1.
Yeah. I think the knockout and the conditional knockout allows researchers to study the pathway in a very tissue-specific or organ-specific manner.
Exactly. So, especially the CKO can be tested in organ-specific manner, so that makes it attractive for the scientists.
Right. And the other thing, Mark, you mentioned is that besides, say, the peptide-based agonists, there also seems to be a trend shifting towards a small molecule. So for one reason, it's more from a patient compliance.
It's the oral availability.
Right.
So like you move away from the injectables to an oral available product that is easier to use.
Yes. And so in this case, how you see the humanized mice can sort of come into play in this setting?
Yeah, we, like Cyagen offers like humanized models. Our human GLP-1 receptor model is one of the examples here. In our model, we replace the endogenous mouse gene of interest with the human counterpart. Yeah. And in this case, we insert the extracellular domain of the GLP-1 receptor and also the 7 transmembrane domain. Mm-hmm. So that is all human. That we only maintain, like in the untranslated region, the mouse part active, right, for signaling purposes. But for the interaction of the drug, of the compound, now with the human gene, or in this case protein, we have now a completely human situation that we can use, or the scientists can use for testing of the drugs.
Right. So by humanizing the GLP-1 receptor, you increase the chance of more translatability, because for some small molecules, they're only bind to a certain residue that was absent in the rodents, but only in humans.
That's exactly the place. So like the mouse GLP-1 receptor and the human counterpart, they have a high similarity.
Right.
But when it comes to the small compounds, they are really specific, or usually specific for specific steric sites.
Yeah, conformations.
And there can be the mouse part is not working. So there's there's an example for a tryptophan, for example, in position 30 of the transmembrane region. That is, in this case, targeted by a small compound that is not present in this specific steric conformation in the mouse model. So you would get false negative results for such drugs that you would test that normally would show nice effects in the model.
Yeah, the humanized models are exciting from a translational perspective. But beyond the GLP-1R, some others say GIPR, we also see a lot of emerging targets. When we look at the pipeline from Eli Lilly or Novo, there also seems to be a lot of emerging market. For instance, one of the issues with GLP-1R is not only body weight loss, but muscle. Right?
We lose muscle. So we are working on, at the same time, like on models where the muscle mass is not affected. So you have a weight loss, but you don't have a loss of muscle in this case. That's the beneficial part. And we have a couple of models that are on the development for this direction. Yes.
Right, that'll be the holy grail, right? You only lose the body weight, but you maintain the muscle.
Yes.
Yeah.
Yeah.
So, So most people know Cyagen as an animal model provider, but we also have the CRO capabilities.
Yes.
Can you elaborate on what CRO services we can provide to the researchers, especially on the metabolic platform?
Yes, I can give you like a short overview. Like for such models for GLP-1 receptor and studying diabetes and obesity, we can do all the basic phenotypic analysis, like body weight, food uptake or so. But we can of course do like glucose measurements, insulin. We can measure hormones. We can do the standard for PK and PD studies. We can do all the enzymes, enzymatic panels. We can do histology. And in support of the scientists that request such models or CRO work, meaning we cannot only provide scientists with a mouse model, but we can also do their CRO work in support of an IND or like for clinical trials.
That's really great. It shortens the time you versus have to buy the mice and then to do it at our own facility or find another CRO. We can provide an integrated service.
Yeah, it's a one-stop solution in a way that we offer. And we have lots of positive feedback here. So and we of course we can customize everything for the clients. In this case, there are specific tests needed or required. We can implement that, and we can also design new models for clients, of course, customized models and test them. Just to mention, like we also have an MRI that we are using now to analyze body fat composition or muscle composition. Like this is really top notch and in line what is expected for the upcoming work.
Excellent. Looks like we got a lot to offer in the metabolic platform, right?
Yes. Yes.
Okay, great. It's nice catching up with you, Mark. And I just want to make a final announcement here. The American Diabetes Association meeting is happening late this month, and we invite you to visit our booth at ADA. Thank you again, Mark, and thank you for listening. I hope to see you in the next episode.
Thank you so much for having me.
Okay.
And stay tuned.
Okay, great.
Thank you.
Okay, bye-bye.
Bye.